Good morning, everybody, or strictly good afternoon, and welcome to the next installment of our COVID-19 ongoing series of webinars, which we hold every lunchtime on Tuesdays and Thursdays. So today, I'm very pleased to be able to welcome two world experts to discuss the very important issue of vaccination for COVID-19. I should say that our two guests, Robin Shattock comes from Imperial College London, and Thomas Breuer is talking to us from Belgium and works for GlaxoSmithKline, GSK. So Robin, if I can start with you, is a vaccine for COVID-19 really the only way that we're going to get out of this current pandemic?
It's not the only way, but I suspect it's going to be the most effective way. What may be more frustrating for people is the timeline for that vaccine being made available to the wider public.
Thank you. Then we'll go over to Thomas. Thomas, according to Wikipedia, my source of information for this mainly, it says that there are 157 vaccines in development worldwide. So of those, have you any thoughts about how they're going to be developed and which ones are going to be the winners from those?
It would be too early to make that kind of prediction. However, I think what is important that there is a worldwide effort, universities, startups, big companies all work together on a solution. It is good actually that we use different technologies, messenger RNA, standard protein-based vaccines, adenovirus-based vaccines. So it's good that there are a lot of different shot on goals, because vaccines, as we just heard, is probably the best way out. I cannot predict who is winning. I hope several of them will actually win, because if you potentially think that you need billions of doses to vaccinate large parts of the world population, you need more than one manufacturer to do that.
And is it true that Glaxo are collaborating with the French company Sanofi? And I think that's unparalleled, isn't it? That two competing companies are working in conjunction. Just explain how Glaxo and Sanofi are working together.
Yeah. So GSK early on decided we will not develop our own vaccine. However, we will make our adjuvant technology available to potential partners like Sanofi. We have other partners, 3 companies in China, another company in the US. And what we do is we essentially put 2 pieces of the puzzle together. We have an adjuvant. An adjuvant is an accelerator or booster of the vaccine, and we combine this with the protein antigen of the vaccine, and therefore create a more potent vaccine. And in a pandemic situation, the advantage is that when you do that, what we have seen in the past flu pandemic, that you can have what we call dose sparing. So you combine an adjuvant with an antigen, and therefore can operate per dose with a much smaller amount of antigen, and therefore can produce many more doses. So in the flu pandemic, due to this adjuvant technology, we were able to produce 4 times as much flu pandemic vaccine as we could have done without the adjuvant. And we are making this technology available to partners which are normally our competitors. That's correct.
Right, that's nice to hear. Thomas, these programs are by health professionals for health professionals, but I think there will be some confusion about all the different types of platforms or vectors used to get the immunological response to the various vaccines. So your vaccine is a mRNA, an Imperial, so I understand. Could you just explain briefly the different types of platforms that people work with?
Yes, so we're working with an RNA platform that's based on nucleic acid. I think people are more familiar with DNA that makes up your chromosomes, and RNA is essentially the message that's translated into proteins. So like the blueprint, if you like, that's used to manufacture proteins. And what we do is we take the sequence for the surface protein of the virus, and use that genetic code to vaccinate into the muscle. It has to be put into a special delivery mechanism, fat droplets. The muscle cells take it up and they start expressing high levels of the coat protein of the virus. And the immune system sees that as foreign and makes potent antibodies against it. The advantage there is we can make a lot of vaccine very quickly. So in the equivalent of a litre bottle of lemonade, we can make up to 2 million doses. And that's very different to conventional approaches that might need 10,000 litres or more to make the same number of vaccines. The other platforms that are out there, there are viral vectors, and I think people may have heard a lot about the Oxford group and AstraZeneca, who are using an adenovirus to deliver their vaccine. Then there are approaches like Sanofi and GSK and others who are making recombinant proteins. So they're making that surface protein, but manufacturing and using that to inject as their vaccine candidate. And the range of different approaches is really very large. Now, whether all of those 170 different vaccines will ever make it to humans, probably unlikely. And I think we'll see a much smaller number that make it to clinical testing.
We've had a question in from Nicholas Stignan, one of our devoted supporters of the RSM, talking about vaccine nationalism. Do you think that's going to be an issue that the developed countries, rich countries that develop the vaccines will keep them for themselves and not pass on successful vaccines to developing countries, to poorer nations?
I think that's definitely something that we need to be, aware of, and I think globally people are aware of it, and there are some very powerful actors making sure that vaccines should be made available. The World Bank, Gavi, governments, agencies like the Gates Foundation. But of course, to some extent, countries that can develop their own vaccines will be in a place to test them and use them quickly themselves first. So in the UK, the two vaccine candidates that are being developed from Oxford and Imperial will be made available to the UK population initially, because the government will put in the money to make those doses at risk before we know if either of those vaccines work. So in that context, they'll be made available first, but there is every intention to make them available globally. But we need to remember that making billions of doses is very different to making a few million.
Just tell us briefly, and I'll go back to Thomas in a second. Sarah, we did ask Sarah Gilbert if she would appear on this program, but she was there was a problem, so she couldn't. But the Oxford vaccines had a lot of publicity and the link up with AstraZeneca. So what's the difference between Imperial and the Oxford AZ vaccine?
So there's quite a lot of differences. I mean, first of all, the Oxford approach has been already tried in a lot of humans. So they have a lot of human safety data, which means that they can move much more quickly. And that's probably why they've been picked up by AstraZeneca. It's very good to be able to give an initial immune response, but it has limitations in that the ability to reboost immunity may be less good than other approaches. So the approach that we're developing allows you to reboost multiple times. We're often pitted against each other, or seem to be in a race against each other, but actually we're collaborating very closely together, exchanging material, and the two approaches may well be able to be used together, with used in a prime boost approach. So we're not actually trying to beat each other, we're trying to work together and make a vaccine available in the fastest possible time.
So let's go back to Thomas. And Thomas, to prove that a vaccine is both safe and effective, just talk a little bit about that. And we've got a question from Samantha Levin saying, how resistant will these vaccines, once proven safe and effective, how resistant will they be to virus mutation or genetic drift, I think is the word for that. So two questions there, Thomas.
Yeah. Yeah, so normally vaccine development takes a long time. We usually say at least 10 years. So people are wondering, how can we even attempt to produce a vaccine within a year or 18 months, but follow the safe criteria as far as efficacy and safety is concerned? First of all, timelines are accelerated internally, but also externally, because regulators and company working hand in hand in making sure that no week or months is wasted by not coordinating these efforts. The other thing is, when you, for example, try to develop a vaccine against a rare disease and you want to run an efficacy trial, you need years and years to accumulate enough cases to actually prove that your vaccine is efficacious. If you run an efficacy trial in an acute outbreak situation, you will rapidly accumulate enough cases to show that the vaccine is effective. So the specific situation of developing and testing a vaccine in an outbreak situation, as well as all parties concerned working very, very closely together, makes it more realistic to have a closer timeline of a year and a half. Then your second question was, if we have a safe and efficacious vaccine, will it stay efficacious forever? If the virus mutates, obviously it depends on the mutation. Right now there are lots of small mutations which doesn't seem to change the virus fundamentally. Again, to use past experience with our adjuvant technology, for example, for HPV, but also for flu, we could show that we have some level of cross protection. So smaller mutations can still be covered by the vaccine. But if there's a major mutation, like we see, for example, for the flu virus all the time, and therefore the virus or the protein in flu vaccines has to be regularly changed, because the virus mutates so far away from the original one that the vaccine has to be adapted. But I mean, it's early days. There are many institutions around the world who constantly monitor the virus, and should that happen, then we will, as an industry, respond to that.
And the genetic structure of the virus was published on I think the 11th of January, wasn't it, by the Chinese? And how helpful was it to have that genetic map of the virus?
I mean, it helped us enormously, because we were able to go from that code and generate a prototype vaccine in a matter of a couple of weeks. And that's partly because, we're using genetic code as our vaccine. It also means that we have the ability to change the vaccine very quickly. So if there is escape, if we do see resistance against a vaccine, we just change the genetic code to account for that. So it was really useful to make that available. And I think that's what gave all the different groups around the world a real head start for getting the vaccine programs moving.
And people talk a lot about the spike protein that enables this virus to get into the cell. I mean, is that one of the main immunogenic factors, or is it something different?
Yes, and the reason it's the main target is because that's what's used to enter cells. So if you can raise antibodies that block that process, we call those neutralizing antibodies, they will just prevent the virus from infecting any of the cells. And ideally, if they're potent enough and the levels are high enough, they may prevent infection altogether.
So Thomas, coming back to you, there's a question from Stevie Isaacs, Dr. Stevie Isaacs, a child psychiatrist, I think. He says, what's the fastest time ever that a new vaccine against the virus has been developed before this?
I think it's in the magnitude of years, 5 to 8 years. An unfair comparison is the flu vaccine, because the industry is used to essentially exchange the material in the flu vaccine because it adapts. But what is currently ongoing, more than 160 shots on goal with many research groups around the world, trying to develop a vaccine as fast as possible is unprecedented.
And let's go back to Robin. Dr. Catherine Mandel says, how safe are mRNA and adenovirus vaccines in immunocompromised patients?
So I think that there are some reservations about using the adenovirus in immunocompromised patients because it is a virus. RNA vaccines don't produce virus-like particles. They can't cause problems in immunocompromised individuals. So we wouldn't see that there was any reason not to use those in those populations. Of course, if they're immunocompromised, they may not make such a good vaccine response. So that is another consideration.
Well, there's another safety question here from Anthony Kerry. Unfortunately, we have so many questions in, I can't possibly answer, put them to you, all of them to you. But Anthony Kerry says, is there any risk that vaccinations can enhance the cytokine storm? In other words, I think antibody-dependent enhancement. Do you think that's a risk in this disease, this vaccine?
So theoretically, that is definitely a risk. We think it's a very low risk, and it's a risk to all the vaccine programs. So people are looking at that extremely carefully, trying to, to reduce the chances of seeing that by doing screening in a range of animal models, including non-human primates, macaques, to see if there's any evidence. Of course, if a vaccine works really well and prevents infection, you're not going to see enhanced disease. So ideally, we need a vaccine that prevents any form of illness. If it does induce illness, then we need to make sure that the vaccine reduces symptoms. So of course, that's something that people are going to look at very carefully. We do think that the risks are low, but they can't be discounted.
And that sort of begs the question of what would success look like? What are you trying to achieve with your vaccine, with any of the 150 plus vaccines being developed? What would success look like, Robin?
Well, the ideal vaccine would be one that prevented infection and prevented transmission. That would be the kind of the holy grail. But in the absence of that, a vaccine that just reduces illness, so people don't have time off work, or even a vaccine that only reduces severity of disease, so we don't see fatalities, would be really useful at this stage. And I suspect we may see that some vaccines may be less than ideal, but may be useful initially and may be replaced by more potent vaccines as we get increasing clinical data and efficacy data. But the most important aspect at this time is for us to see the data, for people to be transparent, to show results rather than press releases, so that we can see comparison of these different approaches.
So Thomas, let's go back to you and let me ask a question. The WHO has received quite a lot of flack from the President of the United States, no less. But I think the WHO has been quite remarkable in its work in helping develop a panel, a sort of consensus, and also getting funding in for these vaccines. Is that your impression? The WHO has done a good job?
It's always a tightrope to bring, all the countries around the world under a common umbrella. I think the WHO is doing their best. I think they worked early on with the Chinese government. They made sure that, the genetic sequence was made available. They're working tirelessly since it became clear there was a major outbreak in Wuhan to alert the world about the pending pandemic. They're trying to share learnings across country borders all the time. So I think they're doing the job they're there for. And, obviously things can always be improved. I think once COVID will be behind us, there will definitely be post-mortem, so to speak, on could this have been done differently? Could this have been done better? But I think, to have these discussions in the middle of a pandemic is not helpful.
Yeah. Well, Belgium's been badly affected, like the UK and the US, of course. So coming back to you, Robin, there's another group, SERI, isn't there, that you mentioned it before. I mean, just briefly explain how their role in the development of the vaccine.
Yeah, so this is SEPI, the Coalition for Epidemic Preparedness and Innovation. They were set up after the Ebola outbreak in West Africa to be able to respond to outbreaks. And so they are helping in terms of funding a number of different candidates, pushing them forward. I think, initially they were more figured or focused on small outbreaks rather than global pandemics. And so, they're working hard with a range of other partners, global partners, to bring funding and to, bring global coordination in terms of responding with vaccines to the pandemic.
And it's also an organization which is looking after equality. So they're working closely with the WHO, but also with Gavi to make sure that once vaccine in a scalable volume will become available, that all people around the world can participate from vaccine success. So that's really different from the last pandemic, where essentially the majority of the available pandemic flu vaccine went to the US and Europe, to Brazil and a few countries in Asia, but the large part of the population in 2009 never saw a pandemic flu vaccine. And as a result of this, SEPI was created to have essentially funding mechanisms in place to work on pathogens like COVID-19. So I think it's a real progress compared to the last pandemic, and we have to see where this all brings us in a few years from now.
There's a question here asking about the duration of, I think it's impossible to answer this, but would you speculate on how long immunity might be maintained after a successful vaccine, or is it impossible to answer that right now?
I think it's pure speculation at this point. Whenever we launch a new vaccine, all we can say at the beginning, when we have a new vaccine in hand, what is the efficacy and the duration we have seen in our clinical trials. Then we start modeling, have post-licensure studies in place, monitor the immunological response, and over time gain experience. However, most of the vaccine provide quite an extended time of duration. So I'm actually quite optimistic based on all the vaccines which are currently out there. Robin?
Yeah, I think I would agree, we don't know. We do know that coronavirus infections themselves give relatively short-lived immunity. So we hope that a vaccine will do better than that. I think, one of the dangers that we may, see is if vaccines are rolled out early after a few months of data, we'll know that they're protective for a few months. We won't know whether they give long-term immunity. So there is a danger of moving too fast, and we need to keep monitoring to look at duration. And I think we need to have a fallback that if we do need annual boosting, that we have the, a capacity to do that. I think that's a sensible risk reduction strategy.
So Sarah Bunn says, is community transmission at a level where a vaccine can be tested, or will this challenge or will challenge studies be needed? It's a good question.
So the answer is, again, we don't know. I think in the UK right now, the community transmission has fallen dramatically, and it certainly may be quite challenging to see enough infections to get an endpoint for licensing a vaccine in a rapid timescale. But there are other parts of the world where community transmission is very high. So Brazil, for example, is high at the moment. And I think that everybody who's developing vaccines and are doing clinical trials are looking to do trials in areas where transmission is highest. The infectious challenge experiments is very controversial. We don't have at the moment effective treatment for COVID-19. And so ethically, people debate whether that's an appropriate way forward. And even with an infectious challenge model, you'd still probably be required to do those efficacy trials to get marketing license. So that they're not necessarily a shortcut.
There's some great questions coming in and making my job a whole lot easier. So Dr. Nathan Sinclair, I assume is a doctor, says, does the time frame being given by the government and the media, especially the media maybe, i.e. the end of the year or early next year, does that match up to what the science looks like? So what do you think, Thomas? Are we going to get this vaccine and we're going to get vaccinated by Christmas?
So when I was asked in February, I always said my best estimate is 12 to 18 months. We might do better because, later clinical trials in outbreak settings like Brazil are about to start. So we might have real efficacy data by the end of the year. However, I believe because the efficacy will only be good, as the other speaker just explained, for a few months. So I think regulators always have to keep the balance between the benefit and the risk of a vaccine. So I could see under most optimistic circumstances that at the beginning of 2021, there might be the first or the second vaccine available for emergency use or for use in specific populations, for example, healthcare workers. I can see that, but it's speculation at this moment in time.
What about you, Robin? Am I going to get a dose of vaccine in my Christmas stocking from Imperial?
Not from Imperial, maybe in the first 2 quarters of next year, if things go extremely well. But again, it's getting those numbers to show that it works. And so we hope next year, but there's no guarantee or certainty, A, that any individual vaccine will work, and B, that the data will be reliable or robust enough for it to be licensed. So at the moment, there's a lot of speculation, and we really need to deal with facts and data rather than over-promising and under-delivering.
Well, there has been a bit of that around, I think, especially in the UK. I think we better wind up there. I did want to get on to talk about anti-vax and conspiracy theories and vaccine hesitancy, but I think we would run out of time. And actually those concluding thoughts from you, Robin, were very helpful. So I'd like to thank Dr. Thomas Breuer from Glaxo and Robin Shattock from Imperial for a really great webinar. Just a few announcements. We have another COVID-19 series webinar on Thursday, where Russell Viner and Sarah Hanna will be talking to Thomas Mars about pediatric aspects of COVID-19. Tomorrow night, a bit more relaxing, our presidents, Sir Simon Wesley, will be interviewing Philippe Sands, international lawyer and writer, communicator, broadcaster extraordinaire, at 7 o'clock for about an hour. And just a reminder that we also, again, a bit more relaxed than the scientific webinars, we have a wine tasting session hosted by Jane McQuitty and David Gleave to talk about some summer wines. Who knows, a glass of summer wine might be helpful in this COVID-19 pandemic, certainly psychologically. So thank you so much, everybody. More than 2,000 people logged on to listen to this. Very proud that so many people listen. It will be available online. So thank you, Thomas. Thank you, Robin. And thank you, everybody else. Be safe out there. Beware of the virus. Bye.